Hello, welcome back to another science episode. Let me just check my mic really quickly. Okay, it works. So, hi, I'm Millennium, Isekai'd Mask Knight Empress Scientist, and today we are going to discuss neurons, con- like neuronal communication, cell to cell communication, and also apoptosis in the end of the stream. And then after that, I will do the set number 10 of the jumping jacks later, if I remember. Hopefully we do. So let's go. Regarding so I think I just need to draw this part, right? I'm gonna draw the cell body of the axon of the neuron. Let's do this one. So you have a squiggly line here. No, apical dendrites first. Apical dendrites. Let's see. Here. Okay. You have these squiggles. And they connect until you have the cell body of the neuron. And then you have some side dendrites going over there. But the important part would be down here, the axon hillock and your myelin sheath here, like that. Okay, I think it looks decent now. So you have the apical dendrites here, apical dendrites, the cell body, the nucleus, the basal dendrite, and you have the axon hillock somewhere here. Axon hillock, axon initial segment, the myelin sheath, and the nodes of Ranvier over here. Nodes of Ranvier. And at the end, oh, we need space. Oh no. Around here at the end point, this looks like this, right? So if you zoom in there, you would have, okay, I'm just gonna draw it over here. That end part would be your presynaptic terminal. And then your synaptic cleft, aka the space where they talk, synaptic cleft, and the postsynaptic dendrite, which is like essentially just a new neuron over here. It's the new neuron body, like that. So you have a oh no, I should have used red here. Presynaptic terminal. Oh, stop. It has to be a smaller eraser. There you go. This is the presynaptic terminal. It's presynaptic because it's before the synapse. Presynaptic terminal. And then synapse or synaptic cleft. And then postsynaptic terminal. Alright. Now that we drew them, postsynaptic synaptic cleft. This whole thing is the synapse. Alright? So first, let me just draw it. The neurons would receive incoming signals through the apical and basal dendrites. And then those signals would be from different neurotransmitters. Take note, that's what's happening here also. There's a different presynaptic terminal over there. So they receive the signals from the dendrites, like apical and basal dendrites, using neurotransmitters binding to certain receptors. And if the sum of the excitatory inputs would surpass a certain threshold, then the neuron passes the fire, like passes that signal to an axon potential, passes it on there. So the cell body would be the one that will process that and integrates or adds up all the incoming signals. And your axon hillock will determine whether the threshold is reached. So this one. So again, you receive signals from the apical dendrites, apical and basal. The cell body would sum it all up, process and integrates. Axon hillock checks whether the threshold has been reached, and when the when you have an EPSP, you have when excitatory postsynaptic potentials depolarize the membranes to about negative 55 millivolts, your voltage gated calcium channels open. So what does that mean? Well, normally you have a certain value, right? But then if you depolarize it enough, aka make it less negative to at least around negative 55 millivolts, then that means this EPSP is enough to cause your voltage gated calcium channels to open, Na plus opening. So we need more space for this. I'm gonna draw that one. One moment. We just need to erase it all. There. I hope we if I can make this smaller in a circle. But no. Oh no. Yeah, let's just erase them. I'm gonna draw the sodium channels now. Sodium channels, because I said once the EPSP reaches negative 55 millivolts, then your voltage gated calcium channels sodium voltage gated sodium channels open. All right, and the way and how that looks would be, let's see. Step by step is this one. You have a resting state. Okay, here you have a resting state, and then you have a depolarized state and an inactive mechanism and repolarization of the channel again. All right, so you have the resting state where they are still closed, the channels are still closed, like this. Plus, 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 plus. And then you have your, let me just zoom in the page really quickly. You have your pluses here. Your voltage gated calcium channels are, since it's positive, it will be near the negative side. And regarding this membrane, yeah, that's just your cell membrane. Now, let's see. Monomer, it's a monomer. It's a single polypeptide with 4 domains, 1 to 4. Each domain has 6 transmembrane segments, and S4 is the voltage sensing segment. P segment forms the pore. Hydrophobic inactivation segment acts as the gate. What is that? It's this one. This is your gate, all right? Regarding this, the S4 are all lowered and the gate is closed because the membrane is at the resting potential, at resting. So outside is still positive, inside is more negative. But if you depolarize it, aka some stuff inside became positive, you gain a lot more positive here until about negative 55 MV, then this lifts up. So I'm just gonna draw a different, then it gets lifted because the signs are switched. So now it wants to be up there. and your gate is now open, which would allow sodium to go inside. More sodiums would then flow inside. So more sodium flows inside, and there is even more depolarization happening. And how do you reset that? Well, at first you have the inactive mechanism, right? And that inactivation gate blocks the flow, aka, remember this gate would block the flow, so that there is one way propagation, and those sodiums can't go back up because of the gate. And then you repolarize the channels using the membrane using your K plus channels, the K plus, because as the membrane, it looks like this again, like this, like that. So once it depolarizes to a certain degree, you need to repolarize it as well. So that's why the K plus channels will open. So now like that, and then your potassiums would go out just to reset it. So that's how you reset it. So again, going back to the big drawing earlier, where I'll just draw it, it's fine. Earlier, negative here. So again, going back to the drawing earlier, you have the you have the cell body here, dendrites. This is your nucleus. This is your cell body, the soma, and then your axon, myelin sheath, axon hillock, and then your myelin sheath, and then your nodes of Ranvier, and this is the end. And then you have that's the presynaptic section, presynaptic neuron. Now this whole thing is the presynaptic neuron. And then you have the dendrites of an incoming postsynaptic neuron. And then you have your synaptic cleft. So what happens again? You have the flow signals coming in from the apical and basal dendrites. Your cell body would integrate all of those signals, and the axon hillock would check if it was enough to cause to reach the threshold of -55 MV. If yes, then your sodium channels open, which would allow sodium to flow in and causing further depolarization. And that's all or none, aka that action potential will propagate down the axon and jump like that. The myelin sheath, who makes that one? Well, it depends. If it is the PNS, peripheral nervous system, wait, no, yeah, it is, right? Central nervous system and peripheral nervous system. If it's peripheral nervous system, it is the Schwann cells that make it. But if it's CNS, then it's the oligodendrocytes who make your myelin sheath. Again, Schwann, PNS, oligodendrocytes if CNS. So the action potential now jumps between the nodes of Ranvier like that until it reaches the presynaptic terminal, where the voltage gated calcium channels would open. All right. So if we want to zoom in there, I'll zoom in a bit, but that's what happens, because just so we keep it in the zoomed out version first. So once it reaches the presynaptic terminal, voltage-gated calcium channels open. There is influx of calcium, and that would lead to the synaptic vesicle fusion, okay? And your snares. That would lead to, the snares would like flow like that. That would lead to your neurotransmitter release into the synaptic cleft. Neurotransmitters would diffuse there and bind to the receptors on your postsynaptic neuron. And now it depends if it is an excitatory or inhibitory effect. So, excitatory effects would be if it is glutamate, and that would, let's write it, excitatory, and wow, the positive neurotransmitters. Let me just draw it out here. Excitatory neurotransmitters. That would be like your glutamate, which would lead to activating the opening of ionotropic receptors, aka your Na plus and Ca2 plus would influx, going inside. So further EPSP potential, excitatory postsynaptic potential. But if it is negative, aka inhibitory, then that would be by like something called GABA. And what happens? GABA receptor would open, allowing Cl minus receptors to open and flow inside. So yeah. And if the postsynaptic cell reaches the threshold, it will just propagate it again. If not, then the signal will dissipate and stop. Note, synaptic vesicle recycling will occur via like endocytosis. So again, for the flow, signals from the cell body would come in. From the dendrites would come in. From the dendrites, and then if that signal will be integrated by the cell body, axon hillock checks if it reaches negative 55 MV. If yes, then it will flow and jump. If yes, then your sodium channels would open, and then it would go up, allowing it to go inside. Then you have your gate to stop it from flowing back. And then how is it reset? Well, your K plus channels could open, and then K plus would go out. It would repolarize it. Then just looking at what's happening right now, it will flow until you reach the presynaptic neuron. If after the action potential reaches your presynaptic terminal, the calcium channels would open. Calcium would come inside, and then like more calcium influx. Synaptic vesicles would be triggered, and they would then lead to neurotransmitter release at the synaptic cleft. Now, if we want to zoom in at the transmission process, a little bit of a zoom in time. Here, we're gonna draw it like this. Here, 1, 2, 3, like that, and then like this. So you have the axon of presynaptic neuron, and postsynaptic neuron ourselves. Same. This is the axon terminal. And then you have the vesicles carrying some neurotransmitters, NT, and after they attach at some point there, NT will flow out and go to the receptor here at your postsynaptic cell. Receptor. So this is the flow of how they talk. So action potential or electric signal. Hello Elmore, welcome back to the stream. We're talking about how the neurons talk to each other. So for action potential, they travel down the axon of the presynaptic cell. Here, presynaptic cell, to the axon, what was it? Travels down to the axon of the presynaptic cell to the axon terminal. And there is arrival of the action potential, which triggers the calcium channels to open. Calcium rushes in, calcium binds to the synaptic protein, and then you would trigger some vesicle fusion here, and then your neurotransmitters can flow to the receptor of your postsynaptic cell. Yippee! Indeed, that's how it happens. Now we will zoom in one more time just to draw how those like clefts connect, but it'll be a bigger drawing like this and like this. There. So you have an early endosome. Number 1, newly formed synaptic vesicles will uptake. Oh, Tonikawa, I haven't watched it yet, but it is on the My Anime list, and I am planning on yapping some of the My Anime list currently watching and completed watching and reading, or plan to read in a different like stream. But yeah, that one is on my plan because that one looks pretty. Now for you have your newly formed synaptic vesicles. It will take your NTs. So let's say there's a newly formed vesicles. It will get neurotransmitters via the hydrogen pump, right? Via the hydrogen pump. Now the vesicles with your NT, vesicles with neurotransmitters, will mature and activate or just stay in the active zone. And the note, you have your like this. Oh, by the way, for the H pump, I'm just gonna, alright, there's an empty. Using the hydrogen pumps, it will get more neurotransmitters. Now you have the snares here. Once they, what's this called? Once they mature. And then the vesicles will dock. Vesicles dock at the active zone. So this is the active zone. They will dock there. And after that, take note, you still have your neurotransmitters. Hello, deleted. Welcome to the stream. Oh, and you might be new, so I'm gonna do the introductions again. Hi, I'm Millennium, Isekai'd, Mask Knight, Empress Scientist, and sometimes I do Pal World, Pokemon, and Science. Yes. Regarding Anime Tonikawa, Mizumi Navala. Okay, no problem. Hello, Mizumi. Welcome to the stream. And we are learning about the neuronal flow right now, and how the neurotransmitters assists in that. Yeah, for the neurotransmitters, they will, so after it docks, the ATP, you have your ATP, right? A retired Vtuber. Okay. Congrats on the retirement, and I hope you're enjoying it. ATP. Okay, I got it. But do you go by Mizumi or Nabola? Your ATP would, what's this? You can't read it. ATP prepares the vesicles for the calcium triggered fusion. Okay. ATP. The snare complex is also priming. Then after this, neurotransmitters. Clervy Valenhart. Okay, no problem. So which one do you go through usually? Or is it just deleted or Mizumi or Clervy? Nice names, good name choice. You have your transmitters there. the action potential arrives, AP arrives. So calcium channels would open, would open, and then calcium influx. The calcium would bind to the synaptic protein. Hello, Decipher. Welcome back to the stream. Neurons are gonna be talking today. Yeah. So once your action potential arrives, calcium channels open. After it opens, then where is it? I have to move this one. After your calcium channels open, the calcium can come in, and then after the calcium channels open, they come in, they would attach to your synaptic proteins. Okay, this one's the cool part. Calcium attaches to the synaptic proteins, and that would trigger this fusion, okay? This fusion with the membrane, aka exocytosis. So that's how your neurotransmitters can then go out. Oh yeah. Okay, that's how the neurotransmitters go out. Yeah, there is no such thing as a sleep schedule around here. That's why time zones are cooked. No, hello George. We were talking about how time zones do not exist and how the neurons are actually talking to each other. So yeah, it could be AM, it could be PM, it could be noon, it could be midnight in different parts of the world, the galaxy and the universe. What matters is we are still breathing. Yeah. So that's the thing. Once the calcium would bind to... Once the action potential arrives, action potential would... Here in this terminal, it would induce the opening of the calcium channel. Calcium would flow inside. And then it would bind to the synaptic proteins and cause the binding of these vesicles there, and then boom 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 boom, neurotransmitters can go out. Now the vesicle that we finished using is now empty and it can just be recycled. So that's how it all happens. And I'm gonna talk about a very very specific example, such as using acetylcholine as the neurotransmitter. Acetylcholine, like this. The ACH is the neurotransmitter. Oh, side note, this is very funny because this is in the Bio Ink stream yesterday, or what is yesterday? A few hours ago? 12 hours ago? I don't know. We were talking about Parkinson's and the brain disease is just killing the patients, and I was like, tisk. So yeah, that's why we're discussing the brain and stuff right now. Yeah. So that's how the streams are connected. Lore. So ACH, now we have, hours ago. Thank you for confirming. I was like, I have no concept of time right now. We are just floating around in the universe like a wanderer. Is that what it's called? We are an Isekai'd Masked Knight Empress scientist. It works out. It's still in the lore. So yeah, ACH, neuro, and this is happening in the neuromuscular, sorry, I'm fangirling over Neuro and Vidal's adventures. Neuromuscular junction signaling. NMJ. This is where an action potential in a motor neuron triggers muscle contraction through acetylcholine release. How your biceps would happen. Yes, and then Elib's new music video, Boom, is amazing. I'm just gonna write it like this. Boom. Boom. And it's reaching like higher numbers compared to life, so that's crazy. Yeah, so for the neuromuscular junction signaling, this is how an action potential in a motor neuron triggers muscle contraction. I'm going to draw an arm. This is the muscle, and this is 1, 2, 3, 4, 5 fingers. Wow, it looks like a foot which has a very big bukol. Anyway, so we're gonna draw it now. This one is your presynaptic neuron, motor neuron. This vesicle has the ACH. Calcium would come in. Okay, the action potential comes in. Calcium channels would open. Calcium comes in. Your vesicle attaches and releases the ACH. And then ACH binds to the postsynaptic. Here. ACH binds to here. Yeah, Vito still has a lot of programming to do, especially neuro and LF security. Being famous and online has a high potential of breach. I did see some clips of him. What's the term where the DDoS attacks? He was getting attacked like numerous times, especially from like California and Japan IPs. It was like, whoa. And he was doing that all live. It's crazy. Yeah, programming. So your acetylcholine will attach there. It binds to the postsynaptic nicotinic acetylcholine receptors. And those would open then. And guess what? There would be NA influx, and 2K efflux. 3 NA and 2K efflux. Then you cause a depolarization of the muscle membrane. Depolarization, aka from more negative to less negative. Depolarization. That's why it's kind of being a bit positive, because it's being less negative. Depolarization, and if the excitatory potential reaches a threshold, your sodium channels open and trigger the muscle action potential. The action potential would then go through the muscle fiber, which travels to what? Through the T tubules, like this. The T tubules looks like that. T tubules. Picasso, yes, this Picasso level drawing. Picasso. The T tubules would be where it flows, and then it would activate the DH, the Doppler receptors, triggering, like now you have the sarcoplasmic reticulum, triggering the ryanodine receptors to open. What happens when that's open? Is that supposed to be an arm? Which one? There's a lot of questionable things around here, but indeed this is an arm with the muscles. So strong. That's the one I keep drawing. No, not drawing. Where is muscle? I'm gonna try to find the muscle emote. Look at this. Whoa. There you go. It's supposed to be that one. That's my attempt of the muscle emote. Is it? Wow. No, it's so far. 1 second. Let me just try to draw it. This one. Okay. Now it looks like an arm. Right? This is muscles. That's the neck. Yeah. See? Yeah, now this one. Oh, yeah, that's true. This one should move there. Okay, and then you just need to flip around the arm. Okay, now anyway, this one looks much better. Good thing we have a source now about how it should look like. Now, for the T tubules, okay, you have a motor neuron. ACH. Motor neuron would be the neuron that talks to your muscles. Acetylcholine gets sent out, would further depolarize the thing, flows through the dapper, and then which would lead to your ryanodine receptors to open. Calcium will now flow out, a lot of calcium, and then they would bind to the troponin and myosin, which will do the the circa, aka the tightening, no, contraction. That would be your muscle contraction. Myosin binds to actin, and using ATP, you would get muscle contraction. How do your muscles, including the heart, how would that relax? Your circa pumps would restore the resting state. Okay, that's also why, for example, you have botulism or myasthenia gravis, where they attack these receptors. Myasthenia gravis MG attack these receptors. It's an autoimmune attack on the acetylcholine receptors, which lead to muscle weakness. How does it lead to muscle weakness? Because if your motor neuron, aka even if you try to tell your muscle to contract, if ACH cannot bind to the receptor, then you don't get the flow of this to the sarcoplasmic reticulum and calcium flowing out, and you can't get this. Can't get muscle contraction. So connecting the, what's this called? The connection between neurons and cancer. What is the connection? Neurons would play a crucial role in cancer development, especially. Let me try to sum stuff up. Neurons, here. There you go. So neurons and cancer. Neurons and, whoa, why is it green? I didn't change the color. Neurons and cancer. Okay, so neurons play a crucial role in cancer development, especially in the tumor microenvironment, because neurons release, remember, they release neurotransmitters which can promote the cancer cell proliferation, survival, and migration. And in cancers like glioblastoma, glioblastoma, and etc., the cancer cells can hijack the pathway that we were talking about earlier and invade the immune response. So what is more of if they are screwed up like that? So you want them to be okay. You want it to be okay. Yeah, thanks for the clarification. But also, what is a xenograft? Remember Xenogard in Pal World? That's why it's a cool word, and whenever there is a Xenogard, I try to catch it. But there is something called xenograft. Xenograft? Yeah, there's something called like that. It's a research model where, let's say you have an animal. Oh my goodness. Anyone guess? Nope. Guess what this is? Like this. Okay. So that's the animal that we have. You put human cancer cells on here. You implant it. It's almost that, but it's the thing that Cat Von Feline eats. I don't know how to draw a mouse. Jerry. I will just say this is Jerry. It is indeed a mutated cat and rat. This is Jerry from Tom and Jerry. I saw that video before. It's like, do you know Tom and Jerry? Yes, Jerry is in the room. That's how they explained that a mouse was in the room without saying that a mouse was in the room. So yeah, you have Jerry. You put human. Yes, it is Ratatouille. You put human cancer cells on an immunodeficient mice, aka a mouse that has lower immune system. And usually that is done to study tumors and drug responses. And here's the thing, why are they immunodeficient? Because you can't just pick up any mouse or rat, especially in the subways or in your backyard. No, that's not gonna work. These immunodeficient mouse, they are, if somebody here doesn't know Tom and Jerry, he ain't human. That's true. Or they're from a very different generation. Yeah. So you put you use immunodeficient mice because those mice they do not have functional T cells. And that would prevent immune rejection of the human tumors. Because remember, T cells from the globin stream, they are a part of the immune system. So if they don't have functional T cells, then the human cancer cells are allowed to grow inside the mouse, and they can closely mimic the progression of the cancer. So next is, so again, review what is a xenograft. It is a model where you put human cancer cells on immunodeficient mice, plural or singular. It depends on how many you want. But you use those because you allow the disease to progress because your mouse model lacks functional T cells. Okay, beautiful. Now, how are sodium channels? Not the meese. I know English, bro. English. The English language. Masseuse. So how are sodium channels? What does that end? There you go. Channels. How are they related to seizures and epilepsy? That's a good question. So here's the thing. Remember, sodium channels, they are the ones that open and close depending on the action potential propagation, right? Is when the signal comes in a little bit, sodium channels will open depending on if they reach the negative 55 MV threshold. If you mutate or if they are mutated, if your sodium channels are screwed up, then that can lead to super excitability, which causes the seizures and epilepsy. Because if the sodium channels are open for too long, take note, your sodium can just go in, and go in. That could lead to the neurons just firing like the rocket launcher, like how I use the rocket launcher in Power, just boom, 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 like that. And that would lead to the uncontrolled neuronal activity in epilepsy. I think in Filipino, just the shaking and the and stuff like that. So yeah, and normally your sodium potassium pump, ATPase, would restore the ionic balance of the action potential by pumping 3Na plus out and K2K plus in. I want to make sure that the direction of this one is correct. The sodium potassium pump direction pump. This one is sodium out, 3Na out, 2K in. What is it? So if we get epilepsy, well, I don't know if your neurons are happy. They're just shaking uncontrollably. They are just they're not the ones shaking uncontrollably. You're the one shaking uncontrollably, and it's because the stuff happening to your neurons are being uncontrolled. Are not controlled. They're just like that. Because it's like your sodium channels are open 5 ever, and then your sodium will just keep flowing in, and action potential would not end, keeps on getting sent. So that's why seizures would be like that in epilepsy. Now, potassium channels would mediate the repolarization, aka back to a little bit negative, by allowing the K plus to go out and return to the resting. So that's why if any of these are disrupted, then you get your seizures. Question: let's say you have a cancer caused by a gene mutation. How would we knock out the gene? What do we use? keyword CRISPR, clustered, regularly interspaced short palindromic repeats, because the Cas9, you have this, and then you have the guide RNA. You use it to create double-stranded breaks on your DNA and in the target gene, and then you can just CRISPR, and then you can just either put something else or not. Yeah, it depends. You can also use RNA interference, using small interfering RNA or short hairpin RNA to silence gene expression. How? Because you degrade the mRNA. And you can also use adenovirus-based gene therapy, aka you deliver CRISPR or RNA into the cells by specifically knocking out the oncogenes. Note, adenoviruses do not naturally cross the blood-brain barrier. They cannot cross the blood-brain barrier, but they can be modified to do so. So for the second phase of this one, we will move to, whoa, what is happening to that marker? We will next discuss apoptosis. So, anyone guess what apoptosis means? The A word. Whoa, that's too big. Better. Apoptosis. That means programmed cell death. Programmed cell death. This one occurs, Pop-Tart, yes. This one would happen either due to like a pathological or physiological stimuli. Pathological would be like viruses or DNA damage or hypoxia. Virus could be cytotoxic T cells, which would induce the, what's this called? Apoptosis. Hello, Abhishek. Thank you. Oh, introductions. Hi, I'm Millennium, Isekai'd Mask Knight Empress Scientist, and sometimes we do Pal World Pokemon, studying while on a treadmill, heart rate while catching shinies, and sometimes I do the introductions in the very beginning of the streams in a weird way. Like for example, I might say, magandang gabi bayan, ako po ang 9th member ng Bini Millennium. Or I randomly say, hi, I'm Bini Mika, or hi, I'm Bini Stacy, hi, I'm Bini Joanna, and all of that. Yeah, like whatever just pops up in my head at that moment, it just. So for the pathological, it could be a viral infection, could be DNA damage, maybe because of your P53 being screwed up, or hypoxia. But for physiological conditions, that could be normal biological processes. That might be the embryonic development or immune system regulation. Okay. And don't mind the fact that the heart rate right now is like, this is a resting heart rate. Why is it 100? You know? That's why studying is also a workout, because you work out using your brain, the muscle. Yes. So regarding physiologic, it could be embryo, immune system, or tissue homeostasis. But here's the difference. Apoptosis versus necrosis. Okay? I'm gonna write it in big letters also. Necrosis. Why do I think it's like Necrozoma? Is that a Pokemon? I think so. Maybe. Or a pal. I don't know. It's all mixing right now. So the difference between necrosis and apoptosis is one is controlled. Greetings, Sheriff. Welcome to the stream. We're talking about controlled cell death versus the cells just dying. Apoptosis is controlled, meanwhile necrosis is uncontrolled. Red, uncontrolled. It's always pathological, and it's a sign of pathology because it indicates that the cell is beyond repair. They're goners. They're goners. So that might mean that cell has trauma or physical injury because they were crushed, or because of toxins like poison causing the cell lysis, ischemia, aka oxygen. This one, listen to this part: oxygen deprivation leading to infarction and heart attack or stroke. That's why if once you get a heart, well, hopefully you don't, once a character gets a heart attack, you try to revive them immediately, because if you don't deliver the oxygen to the brain, then that's the tough part, because your brain really needs the oxygen, and the other parts of your body, of course. So yeah, that's why it's like CPR, boosh, 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 that one. Oh, and severe infections too, because like in tissue necrosis, like gangrene. So that's the main difference with apoptosis and necrosis. I think in a character it would be the difference of they just die. I think if it's, let's say you have, yeah, the sound effects, it's like high Q. So for apoptosis, let's say you have, if it is time for the cell to like just go bye-bye, then I'm thinking of a character right now. Let's see. Not a character. Let's think of, oh, there you go. So for, the skin. Think of your skin. So shedding of intestinal epithelial cells and skin cell turnover. You know that your skin, the skin cells right now on your arm is not the same one in a way when you were a baby, because that's why when you itch it and all that, some of those would come off because you have skin cells turnover. So that one is a bit more in the apoptosis range, because you need those to turn over, to switch around. But necrosis is more of, what was it? Let's say you get a very bad bacterial infection, or you get poison, or tissue necrosis. Like gangrene, I think, is the best example for this. Let me just double check the gangrene itself. This one is the death of the tissue caused by infection or lack of blood control. So see that one, it's more very pathological. So that's the difference. Uncontrolled versus controlled. One would be, I think this one would be more of a seppuku. I think it's like, no, they would choose when they'll like move on to a different world. Necrosis is just, it just happens. Oh, hello, thank you very much. Yes, more science. Let's go. Yeah, that's why we're discussing apoptosis and necrosis. Science. More science, especially in the next stream. That one, the development stream, I don't know how long that'll take. Yeah, whoop. Thank you very much again. So this one, I just want to clarify the DNA. For example, DNA fragmentation, because that's very, very important. Okay. Now let's go with, yes. Oh, that is so. I'm just looking at the color. So that's how it looks like. That's how it looks like. It's rare that I see that one. The super chato. This one is the control, apoptosis, and necrosis. Ooh, okay. I think there was a question about the difference between them. So this one will be a good one. Control, apoptosis, and necrosis. Okay, let's divide them into 3. That's a very straight line. Thank you. Look at that. Just, why is it curved? It's alright. That's just angles. So for, let's say you have DNA, right? DNA. This is DNA fragmentation. in gel electrophoresis. Peak. I know, this beautiful Picasso, Van Gogh, the Mona Lisa would say like, wow, amazing. Yeah, this art. So you have DNA fragments. Note that this would be the bigger fragments, and this would be the smaller fragments. Just angle the ruler by let's say 30 or 60 degrees, because if you just look at that, that's the angle of my lines, right? But if you draw this one, it's a 90-degree angle, and this one looks like it's 30 degrees. No, chotomate. This is 30 and this is 60. See? It's beautiful. Look at that. So we're still using math and straight lines. You just need to look at it with your heart that it is indeed straight. For the control, you have your DNA here. It's intact. For apoptosis, it might look like this, ladder-like. Yes, we are now explaining lines. This became a math stream as well. It might become lines because it's due to the caspase-activated endonucleosomes. Slowly but surely, we're degrading them. But necrosis, you get a smear, not very exact, because this cell lysis is just very non-specific. Oh, side note, look at the mini floating math stuff there on in front of the sunglasses. That one's that's why we're also doing some math right now. Mathematics. Okay, so we did DNA fragmentation and necrosis. Oh, nah. So again, mini review. What's that? The cone. Yes, the cone. So cells undergo apoptosis either through extrinsic or intrinsic reasons. And what's the main difference between apoptosis and necrosis? Apoptosis, programmed cell death. Necrosis, they get killed. There you go. That's the difference. Apoptosis is they can choose. Well, it's like programmed. It's programmed. Necrosis, they weren't able to choose. And regarding neurons, just as a mini review of the whole big picture. Let me draw it here. Okay, let's see. You have here. 1 second. I'm just finding the right page for it so I draw it properly. Anyway, you have your dendrites, your basal and apical dendrites. And then you have the cell body like that one. And then this looks like a Pokemon or a pal. I don't know. What's that? The octopus pal? And then you have the axon hillock and you have the myelin sheath, nodes of Ranvier. Okay, you have and then the end part, this one. So you have signals. I'll label it again. This is the nucleus. These are your dendrites. Oh, and you might have basal dendrites also here. Basal dendrites. I know this is what happens when you have to look at a lot of these stuff. Ever die? No, watch out. There's a couple more coming. But don't worry, some of them will be question and answer on a magical wheeled thing. Look at that. Yeah, and there's the human like that. And then you run zoom. Yeah. That's the basal dendrites. And then you have your axon. Whoop, wrong color. You have your axon hillock, like here. Axon hillock. Oh, good thing we were able to test what is it? The mini Supa from earlier. At least we saw that the CSS works with it. Yeah, we saw how it also appeared in a different way, and it's also coded such that For example, there's like a difference between the different mini chats. Because for example, let's say I type like this, it would show mine as the one with the swords and the crowns. Yeah. So at least we're testing how the CSS works. It works. Yes. Yeah. Now, oh, and how the horizontal also works. So that one, I think, at least it helps it look like a banner. Yeah. And the members would have the mini eye one. If you chat, it would show the eye cover. So that's how it's kind of customized or tailored, so that there's a variety of decoration thingies. Yeah. So for axon hillock, and then their myelin sheath, and then you have your nodes of Ranvier. Ranvier? Nodes of Ran. The spelling is nice. Saying it is weird. Ranvier. Okay. Anyway, you have your signals coming in. If you reach the threshold of negative 55 MV, what happens? Sodium channels would open. Sodium channels would allow sodium to come in. Once that one opens, sodium more influx. Rainer. Good one. Very good one. Nice one, Decipher. Very, very nice pun over there. Imagine if it's Berthold Boruto or something. Once you reach negative 55, take note this is going from negative 70 MV to negative 55 MV. So you're depolarizing it, aka going from a super negative to a less negative, aka a little bit positive. I don't understand much. I'm an engineering student, but I am really enjoying this. Thank you very much. It's because I think science and math are just very universal languages which can transcend the universe, just like how lore-wise I am an Isekai'd human. Not a human. It depends on the universe. That's why. And also wearing sunglasses right now because it might be in different times, so it's a little bit like the sun is over there. Yes. So you have negative 70 to negative 55. Then when you reach that, your sodium channels would open. Sodium would come in. There's an influx. And then, I am an art student and this is toasting my brain. Don't worry, look at the beautiful art. Your neuron right now, it looks like a pineapple. No, it looks like a pineapple. Okay, it looks like a pineapple, and then we will have to draw SpongeBob around here. My apologies. This is SpongeBob. Nope, it looks like one of the wood thing in Ed, and Eddie. Ed, Eddie. Yes, no worries. They will just tingle while you're looking at it. So it's an all or none action potential, aka it happens or it doesn't happen. And now your myelin sheath. Take note that there is a thing about myelin sheath. It depends on if it's in the PNS or in the CNS, aka peripheral or central nervous system. Who makes the myelin sheath? If it's in the PNS, it would be Schwann cells. If it's CNS, oligodendrocytes. Yeah, the drawing is too beautiful, so you have to look at it with an open eyes, heart, and ears. The same as how if you listen using your ears for the mic, you either you hear the jets or don't. Don't mind the jets. The jets it's just gonna be there. Now, once you reach that one, the action potential would then jump from different nodes. It would go like that. Take note that the cell body or the soma would integrate it, while the axon hillock would see. Did we reach the negative 55 MB? If yes, yeah, go, you may proceed. Now, once you reach the axon terminal, what happens? Important things happen here. Once you reach the axon terminal, or the action potential reaches the presynaptic axon terminal, your calcium channels open. Calcium goes inside. Calcium would induce your what's this called? The vesicles like this. Okay, I'm gonna draw it over here. Calcium, because this is how it looks like. zoom this in. It's now over here. You have some synaptic vesicles here in the neural, and you have neurotransmitters inside. But once calcium terminals open, your calciums can come in, and it will induce the binding of the merging of the synaptic vesicles with your membrane, and that's how the neurotransmitters like acetylcholine can go out. And then the receiving neuron or muscle cell or fiber would receive it, and then that would trigger more stuff. In terms of your biceps, let me try, or your heart, let's say your cardiomyocyte, cardio, heart, myocyte, cell, cardio, muscle cell, myocyte cell, heart, muscle cell. Your heart is a muscle. And now they would bind there, acetylcholine. Let's say your NT, the neurotransmitters are acetylcholines. You bind there, acetylcholine receptors receive it in your next neuron or something. And then it would just lead to the DHPR opening, and RIRE2 from your sarcoplasmic reticulum would open. Calcium would then go out and attach there to your myosin, troponins, synaptins, and all that. And then boom, you have contraction. But take note, wait, does this learning method actually work? Like trying to teach someone something so that you memorize that topic better? I don't know, but we're hoping it works. It should work. That's a very, very good cutthroat question, straight to the jugular. But yeah, for now, it's more of I just need to learn how to yap about it more and more with the pen and the paper or something, because I just need to practice talking about it, essentially. Well, for a lack of a better word, I need to learn how to yap more about this one. So hopefully it is. Hopefully I'm explaining it. But yeah, this is just a mini review from the earlier one. We have beautiful figures there. The shaking voice, like in the hopefully it works. Yeah, the mental stability. You know, the alternating personalities are incredibly splitting. It's like half of the brain power is on the drawing and half of it is in the learning and all that. But yeah, and that essentially sums up how your neurons like talk to each other. Oh, did I miss the snap vesicles? Snare complex. Oh no, we talked about this one. Yeah. Now for I think I can give a mini teaser about the developmental lesson, because that one, where is it? Notes. The binders. There are a lot of binders. Okay, so now moving on to, it does help. I just need my brain to stop projecting the wave impulses and accept the signals. Okay, you just need to live and breathe it. Yeah. But now we are talking about development. So mini intro to the stream. Wow, to the steam in a bit. Because I think we can still keep going. Let's go. So, about development. Organisms have many stages in how they develop. We are not talking about ships, not the fan fictions, and not developing of the main character and the side characters. No, not that. This is talking about how you go from gametogenesis to, wow, I just wrote over it. Gametogenesis. Let's see. From gametogenesis to fertilization to cleavage to gastrulation. What is sleeping anyway? That's true. You know, if you think about it, as long as your other side of your soul lives in a different time zone, you technically have all of it could be day. And then if you just want to keep sleeping, just transport your soul to a different part of of Earth, then you could have 24 hours of nighttime. Hacks. Very big hacks. Now, after gastrulation, it would be neurulation. These are all big words, but I'll explain it in a bit. You have organogenesis. You have the larval stages. And then you have maturity. Okay? Gametogenesis, what is that? It's the formation of gametes, the sperm and oocyte in sexually mature adults. Fertilization is the fusion of sperm and oocyte, leading to the zygote. My sleep schedule is doomed anyway, why fix it? His last quotes, and yes, that is true. It's not just doomed or in need of fixing. What if it doesn't exist? Just if you close your eyes, it's not there. It's not there. So that's why you learn about development until like cleavage, where you rapid mitotic divisions producing smaller cells. So that would be, let's say you have a big cell and then you have smaller cells like that, and then you just form a lot more smaller cells like that. Gastrulation is if you divide the cells into the 3 germ layers, aka ectoderm, mesoderm, and endoderm, and the formation of the blastophore. Neurulation, big coat, neuro, for the neural tube, and that's the basis of your CNS, the central nervous system. Organogenesis, genesis of organs. You make your organs and tissues through different interactions. And then larval stages would be immature developmental stages. Cheeseburger, welcome to the stream. Yeah, we finished the brain and apoptosis earlier. Apoptosis and necrosis. The mini one-liner summary: brains. The brain uses neurons to talk to other parts of your body. Apoptosis is programmed cell death. Necrosis is your cells are forced to die due to some certain diseases. Yeah, and now we're talking about development, which is a mini teaser. Or, imagine if I just finish all of development right now. Then we won't need that waiting room for that stream, and it'll just be a different one. There's always more. So yeah, about larval stages, you have immature developmental phases, aka metamorphosis is in some species, like your butterfly. Look at that. Okay, honestly, it looks like a butterfly, right? Beautiful. Maturity is when your organism... More toasting, yeah. Maturity is when your organism reaches reproductive capability and completing the cycle. Yes. Okay. So, we can actually talk about the frog. A frog embryo. It is a moth. But butterflies are prettier. But they're both kind of scary, especially if they fly around because of the... I read somewhere that they're... the powder that when they flap, that it might cause blindness sometimes. So that's why I try to avoid going inside butterfly houses. Is that the term? Butterfly gardens. It's a dome with lots of butterflies. That one. It's on the same level as a haunted house for me. So no. But that's ironic too, because one of my favorite VTubers is Monarch, who's literally a blue butterfly. But it's because she's blue and it's nice art. Yes. Okay, so for frog, the frog, they fertilize, and then you get the cleavage. It is indeed, you get the cleavage, where after fertilization, you would just times two it. Morula would be a solid ball of cells. Gastrulation, neurulation, you form the neural plate. Organogenesis, and brain. Notice how I avoid the horror games. Essentially, I don't play horror games. No, I apologize for those who want horror streams. I apologize. I cannot. Well, that is the one. That's why I'm hesitating about the K-pop DH, because although I would really, really love to watch Gnu, with no spoilers, please. I feel like it might be a bit scary. Do you know? No, don't ask questions about that topic. Thank you very much. But yeah, that's one thing I do not want to discuss. The K-pop DH, it ain't horror. Well, hope. So here's the thing. I just need to wait for a little bit for the what's that called? For the trend to be over. Just a little bit. But the song is so nice. It was especially the Tagalog version of it, and the English and Tagalog version is a nice one. Yeah. So again, for the cleavage of the frog, you have a cleavage pattern of unequal. Is it the cat? I don't know. It's similar as to why I will wait for the. No, I don't think it's the cat. I don't know what's the lore about the cat. It's how can I say this? It's in same as the spicy, where I have the tolerance of level 0. That's the same one for the scary games or haunted houses. But what's funny is I got through Jujutsu Kaisen because Gojo. And then I got through AOT because of Levi. So K-pop DH, I might be able to watch it because Gnu. So that's why. I just need to wait a little bit and load and gather and muster up courage. Yeah, it's very, very base taste. And now we have the cleavage pattern of a frog or Xeno plus egg. The first 3 cleavages, they would be. It looks weird like that. Anyway, it would just keep dividing until you get the neural plate and all that. And then you have a thing called yolk distribution, where the animal pole and vegetal pole. Animal pole has less yolk. Vegetal pole has more yolk. Insta erase. What happened to the insta erase? The vegetal pole? Yas. And now for what's this called? Ingression. No, we'll skip that. I want to discuss organogenesis, making the derivatives or like the germ layers. That one's a fun one. Whoa, where's my eraser? Sorry. Here. Okay, there we go. So you have the zygote. You have the blastula, and then you have gastrula. Good. Now at the gastrula phase, you get 3: ectoderm, mesoderm, endoderm. Ectoderm, mesoderm, endoderm, and some germ cells, but that's fine. Germ cells. Ectoderm, what's that? That's the skin, CNS, and neural crest. Mesoderm would be dorsal. No, I don't want to do that. No, actually, you know what? It will make sense. 1 second. Dorsal, paraxial. Sounded like an evolution. Balasula, gastro. They might be Pokemon names. Or pal names. The ones that I haven't found yet. Paraxial, intermediate, lateral, and head. So your dorsal... Wait, let me just circle this. Ectoderm, mesoderm, endoderm. Ectoderm, skin, CNS, neural crest. Mesoderm is dorsal. Your notochord, CH. Paraxial is bone. Intermediate is tube cells of your kidney. Lateral is your red blood cells. And head is the facial muscles. Guess what? For endoderm, they are the internal layers. Your stomach, thyroid, and the lungs. So see, look, you need them all. Hopefully you have them all. Yeah, you have skin, CNS, neural crust, notochord. So ectoderm, skin, CNS, neural crust. Middle one would have dorsal part, and including that includes your kidney, bone, notochord, heart, RBC. Hopefully, there's a big bird there. Hopefully, endoderm is stomach, thyroid, and lungs. And then, for stages of cell differentiation, you have specification. The cells stop dividing. No, let's start with the cells here. Here. There you go. If there are something missing, well, we gotta catch them all. Yes, we do need to catch them all. If something's missing, not just catch them all, you gotta hunt them down. The same as how the PIDFs would really hunt you down if you accidentally kill like some of the humans or pals in Pal World, and you raise your wanted level to and now you're, instead of playing Pal World, you are now doing GTA. In fact, now you have undifferentiated cells, undifferentiated, and then you get speciation or specification, I mean, where your cell stops dividing and differentiates autonomously. And then you get determination, where your tissue differentiates autonomously. Okay? But that's how it works. And now, as for the types of specifications, we mentioned GTA is indeed, it is the goated chaos. I really liked it, like playing it, especially I would think in GTA, disclaimer, you would take the cars and then just like drive through the curb, and then you would hit the street lights. It's like, oh, so cool. In GTA. So autonomous specification, finished, pattern formation, mapping cell maturation. You could use single cell RNA-seq, where the staged, I wonder if there are any cells that have been with you since birth. I'm not sure. Very, very good question. Let's ask one of our best friends, Google-chan. Do I have cells with me since birth? Google-chan, what's your answer? To some extent, certain cells like those in the brain, heart, and eye lenses are present since birth and are not normally replaced. Yes, Clove from Valorant. Who is Clove? I only played Valorant for a bit, so I don't think I got all of their, what's this called? Like, characters. Oh, imagine though if I'm Clove from Valorant. Imagine. Yeah, that would be cool. Oh, her character design's cool too. Like, imagine a blue version though, and she's holding a gun. Longer hair with, like, super long hair with a gun, or swords. But I don't think Valorant has swords. This is a gun game. I did Counter-Strike before, but I kept hiding. My apologies. So, for single cell RNA-seq, staged embryos are dissociated into single cells. You individually isolate them into droplets with barcoded reagents. What else? Cell-to-cell communication, we talked about this. Juxtacrine, paracrine. What is juxtacrine? That one is you need direct contact. Paracrine, it is a little bit, it's still local, but far. Autocrine is you send out your own. Okay, no, I'll draw it. You have let's start with paracrine. Just as a review, you have, this is your cell. It would send out the signaling molecules, and then a target cell has the appropriate receptor for it, and that's how they talk. Now for autocrine, oh yeah, very, very soon I will have to rest my voice. Like, the people who continuously lap for like 6 to 12 hours are amazing. the vocal cords built different. The autocrine would be if you release the signaling molecule and then the receptor is on you. Whoa, I missed the arrow. There you go. And then juxtacrine is, whoa, spelling. Juxtacrine is here. And then you have your signaling molecule is actually attached. So the target cell would need to come closer so that they can talk. And what else did we miss? Macromolecule integrins, what are in solo leveling, arise has swords. I haven't watched solo leveling, but like, what's the name of the character? The main guy character. I keep thinking that it reminds me of Kirito for some reason, but I haven't watched solo leveling. Song Jin-woo. Oh, it reminds me again of An Hyo-sop, the one who voiced Jinwoo. Yos. So integrins, receptors for ECM. I finished the whole Sword Art Online. Not the light novels, but all of the anime and what's that? All of the movies. So is the game of that anime. Oh, I see. Okay, that would be nice. That would be nice. I haven't played it, but swords. Literally look at my design. I just put me in Sword Art Online. Hopefully I won't stick out like a sore thumb. I will even join Knights of the Blood Oath, or I don't know, I just think it's so cool. Okay, now integrins, they are transmembrane receptors that mediate interactions between cells and the extracellular matrix. You can really see the flipping between the setting. It's like you're talking to two different people. One is like fangirling over Sao, and suddenly you flip over to paracrine and juxtacrine and autocrine. That's the mental state we are at right now. It's fine. It's going. It's going. The going is going. When life gives you lemon, make a lemonade. I can't even drink lemonade. It hurts my teeth. So that's why we do mango shake. Yeah. Epithelial mesenchymal transition. Speaking of which, have you checked out the request last time on the girls? What girls last tour? Wait, what? Did I forget something? Remind me again what happened there. You have morphogenesis, ne. Early development, metazoan, sea urchin, finished. Amphibian early, finished. And finish that one. Beta catenin, that one's important. Don't forget the Wnt signaling. Wnt signaling review. Mini review of the Wnt signaling. Essentially, if your Wnt is off versus Wnt is on, okay? Straight line. Oh yeah, very straight. So this is your cell membrane. No, what was it? Is your cell membrane? I'm scared of searching because what if it gives me a spoiler? But what's the title? Is it Girls Last Tour? Is that the manga? What's the genre? I don't remember talking about this one. So let's say you have your Wnt and then you have your frazzled. Girls Last Tour. Okay. Let me just, I'll mark that. It's in there in the chat description. You have your Wnt, and if your Wnt molecule does not attach there, then the destroyer complex, adventure and psychology. Oh, okay, something like that. Okay, maybe. And your axin and your CDK. I'm trying to write this down from memory. Hopefully this one's the right one. Now, those would phosphorylate. If your Wnt ligand is not there, those would phosphorylate your beta-catenin. And those are the phosphates, and would destroy it. That would be sent to the ER for ERAD. ER, associated degradation. But if your Wnt is active, then is your frazzled, and your LRP, Wnt goes here, and then it would be, it's now active. Your GSK and CK would phosphorylate your LERP. The dish would attach to your frizzled, and your APC would be detached from axon, and your beta catenin is allowed to accumulate, and they would go into the nucleus. After they can go to the nucleus, they will go and tell your that they can now make from DNA to mRNA to protein. Boom. Then you get the proteins that you need for, let's say, your cell cycle or your like cell migration or cell proliferation. That one is why your Wnt signaling is important. Hox family, HOX, that's not HAWKS, not the My Hero Academia one, aka the same voice actor as Goju Sensei. Interesting. OMG, wait, after. Okay, I just need to 1, 2, 3, 4, 5, 6, 7 go through these questions, and we finished developmental biology. Oh, that's crazy. Okay, so what are the signals that drive the movement during gastrulation? Interesting. You have key signaling pathways for migration, adhesion, and cytoskeletal, and especially the Wnt. What signals drive the formation of axial cells? That would be the mesenchymal to epithelial transition. If a PhD student wants to study how the yolk influences gastrulation cell movement in the frog embryo, how would you design an experiment? Home stretch, yeah, the vocal cord stretch as well. I will drink water after. If a PhD student wants to study the yolk and how the yolk influences gastrulation, they would play a central role for the cleavage, germ layer, and all that. And the way you would divide the control groups would be Koseki Biju and Koyori and the others are really built different. How can they? It's amazing. Like that kind of training, they must have muscles in there. Here, the yap muscles. Okay, anyway, you have your 3 control groups: control embryo, embryo with partial yolk removal, and what's this called? And then you have, what's the third one? Embryos with vegetal hemisphere enrichment. So the control, the normal, the removed, and the very high up. So that's how you can see the control would be unmanipulated, and they would just count for normal disturbance. If the yolk content impacts the movement, then the embryos with lower yolk would have accelerated involution, while the yolk enriched would have slower internalization. Because earlier we said, if on college there be dissecting frogs, I'll straight won't attend. I don't want any frog ghost on my sleep. Oh, don't worry. We did that too. We dissected. I have experience dissecting frogs, and I think it was fish. There was time it was fish. Formaldehyde and all that. Make sure to wear a mask and gloves, and don't worry. It will just think about how it is for learning. Especially if it's in a very educational setting. Please don't just go Squid Games, and sometimes there are some educational programs who do fish. But the frog is a very good example also. Very, very good example. Oh, I love fish. I have clownfish. A clownfish. Does that mean I would assume you watched Finding Nemo, because that is that fish. Oh, and there is a certain fish who recently debuted. Very, very nice. Very nice model. Oh my gosh. And so fluffy. I kept on like trying to analyze how the ears on Saba's model works, and I think it's slight. It might, if you notice how when her eyes go wide, then the ears go up. Them opening and closing their mouth the whole day. That's the fish, right? It's very calming, especially koi fish, just floating around. But yeah, if the yolk movement or if the yolk content impacts the movement, then I did not scan the QR code. I did not. I was like, nah. Knowing Saba, knowing who Saba is, that it's funny. It's gonna be funny. It might be like the never the never gonna give you up one. Yolk enriched embryos. If in the super removed one, you might see accelerated, but in the super enriched one, you might see it very like slowed down. What are the signals and structural changes involved when visceral endoderm transitions into epithelial? Just the adherence and tight junctions. What signals and cellular properties drive radial intercalation? Many mesenchymal to epithelial ones. How does the formation of rosettes link met? It depends on its shape. And if a PhD student wants to study how subsurface epithelialization precedes radial intercalation during met, how would you test this using Z01 GFP embryos? Okay, Z01 GFP. Sleepy, no, you won't believe in the power, the Nakama power. Believe in the montage, in the training montage. And same as how Naruto kept on saying from literally, I think, episode 1 that he's gonna be Hokage. And now look, a gazillion episodes later, he is Hokage, who spoiler alert, who unfortunately was not able to attend his own coronation. I was like, why? But it's very Naruto coded, of course, to not be able to attend the whole purpose of the show. Anyway, you have Z01 positive cells, and the live imaging would help see it. So that's, oh my goodness. So we did 3, and I literally need to rest the voice. But we did 3. We did, what did we do? 1 hour ago. It was brain, apoptosis, and developmental biology. So I'm going to update the what's this called, thumbnail into developmental neurobiology, because that's the next one. But I will just need to rest the vocal cords first. Alrighty. But anyway, thank you very much for joining the stream and learning more science. And I think that is all for today. See you next time. Bye.